Okay, so let's start. Today, we are going to talk about the some of the ion channels, transporters, and also the neurons and the glial cells. Those are the basic component that in the nervous system, so-called the building blocks of the nervous system. So last time, just that we talk about the action potentials, how that is being generated, and the channels involved, and so on. So let's spend some time to talk about the ion channels and transporters. Okay. So just give you some information regarding like actually how those recording are being done. That as we mentioned, that you can record the membrane potential or the activity of the channels by the so-called the patch cam. So actually, what people can do in the scientists, they use the really fine glass pipette and then patch the membrane. This is the channels, so you can remove it, and then you can measure the current going through. So there are different ways to do it. So for example, you can get the whole cell recording. So that one, just like when you record the action potential at the axon, so you get a little bit patch over there, and then you can measure the differences between the outside and the inside of the cell. What you can also do is actually get piece out. It means that sometimes if you are lucky enough, then you get one of the channels, then you can measure the channel's activity. So how that is being opened and the current being transmitted through the channels. So it depends on how you do it. So this one is the ends of the channels are outside, it's extracellular part, or you can pull it out as the inside part as well. So in this way, you can really measure the channels. Now, during at the neurons, the surface, the axon that we talk about, the action potential, that is due to because of the large number of these kind of channels. So it's not just individual. So in the next slide, you'll see that actually there are different kind of channels. When we talk about different neurotransmitters, then you will see that the different kind of channels allow the passage of the ions. That's how they control the permeability of the ions. Now there are different kind of channels. There are voltage gated channels. So the channels actually controlled by the voltage. That's the changes in the membrane potential. Remember, in the action potential, those are the ones that actually are controlled by the voltage. Now, for example, the sodium channels, calcium channels, potassium channels, and chloride ion channels. So this type of channels are controlled by the voltage. So if there is a changes in the voltage, then in the membrane potential, then those channels will be open, or some of those may be closed, depends on the type of channels. There are also ligand-gated channels. When we talk about the neurotransmitter, then we'll get more on to those. So those channels are actually controlled by ligand. So in this case, for example, the neurotransmitter receptors. So the ligand will be, for example, in this case, glutamate. Okay, glutamate binds to the channels, and the channels will open. Okay. The other one include, for example, calcium activated channels. So if calcium is the secondary signaling molecule. When we talk about the neurotransmitter or receptor signaling, then we'll get into more of those. Okay. There are also cyclic nucleotide gated channels. Those happens in more of the G protein coupled receptors. As some of you might have studied some of this that in other classes, other courses in which the G protein coupled receptors. Now, when we talk about those receptors and the neurotransmitters, pathway, then we'll get into more on those channels. Now you can see that in some of the cases, the channels will be selective, okay? Now in some of the other cases, the channels are less, more selective. For example, in this case, then it allows the sodium and potassium pass through some of those, okay? So it depends on different type of channels. Now I will post the first assignment soon, which is talk about the potassium channels. In the middle of the class, I will tell you a little bit more about that, okay? Now, as I mentioned about the channels, you can measure that with the patch cam, okay? Now, when you measured all of those in a big bulk, then you'll see that actually, for example, in the case of the channels are being opened, you see that there's a current, going to positive, from the negative and going to the more positive. Okay, now those are actually contributed by individual channels. Okay, so you can see that if you measure different channels, you see that they open, some of those open. So those currents are actually due to contribute by all the channels combined together. So if you measure it and combine together, then you see that there's a downward movement of the sodium ions. If you combine all together, then you can see a very deep, movement of those ions. Now that's the time that actually happened during the action potential, in which that when there's a depolarization, the sodium channels will have a much higher probability of opening. Okay, so that means that that those currents are contributed by all those channels at those surface. Remember what happens in the action potential, that's contributed by all those channels. Similarly, there are also potassium channels. Similarly, when they open, they will open up, and then that will be different current on the opposite. Okay, so when this is during those action potential, those will contribute to the current of those potassium movement. This is the same as in the potassium channel, sodium channels. When there's a voltage change, if those are voltage gated channels, when there's a membrane potential changes, then the probability of those channels will have a higher probability to open. Okay, so those channels combined together are responsible for the changes in the currents. Okay, so it's not just individual, it's a group of those channels together. Okay, so that also, the channels actually contribute to the permeability changes. Okay, so here is one of the example of the potassium channel. This is in bacteria. The reason why behind is actually the bacteria potassium channel is the most simple one, in which that there are 4 polypeptide chains that actually form the pore, in which allow only the potassium to pass through. So when we talk about action potential, it is really selective because of those channels only will allow potassium or sodium to pass through. Okay, so that's the reason behind that. The ions are being really selective. Now, in some of the other channels, they may not be as selective as this. So it depends on which type of the ion channels. And when you talk about some of the ligand-gated ion channels in the neurotransmission, some of those are actually not that selective. So some of those are selective, some of those are not that selective. It depends on which type of the channels. Is it okay? Now, apart from the channels, there are different transporters as well. For example, one of those we talk about the ATPase pumps, in which the sodium and potassium pump or calcium pump, that use of the ATP as the energy in the sodium and potassium transporter, pumping in potassium and then pumping out the sodium. So that's the basic that during the in the action potential that store the changes in the ions. There are also other pumps, for example, in the calcium pump that also use of the calcium and then exchange it with the proton. Okay, those are the energy. Now, as I mentioned before, those pumps continues to work and store the potential energy across the membrane. Okay. Now, in those that can use it for other transport against the concentration gradient. Okay, for example, in this case, sodium and calcium exchanger or chloride exchanger, the proton ion exchanger, and also some of the neurotransmitter exchanger. Okay, so you can see that those pump actually pump and generate the potential, the concentration gradient differences, and those energy can be used to pump other molecules against the concentration gradient. So that's actually a very important aspect, particularly in the neurotransmitter aspect, in which how that you can store the molecules against the concentration gradient. Okay, so that's the very basic. This is same as the case that I mentioned about, in the case of the water tank in the building. So can you tell me, for example, why it is good to have this kind of system? Anyone? Pretty quiet today.
Maybe to affect, to give more type of diversity of responses.
Close. You are a little bit close, but now usually the ATPase pumps will be a little bit more energy saving. It will be a little bit more complicated, the structural wise. So one of the good things is that actually, if you have one type of pumps, and then those energy can be used by simpler transporter. So all the transporter can make use of the same the potential energy. Just like what happens in our society, we all use the most common form of the energy is the electricity. Okay, but all utilities, all other components use the electricity for different purpose. For example, to power computer, or to boil water, or to power air conditioner. But they all use the same system. Yeah, so the pumps actually will build up the gradient, stored as the energy, and then all the transporter or exchanger can use it. Yes, that's as the energy. So it's just like common form of energy that can use across different molecules. Yes, pretty good. Okay. Actually, you see more of those examples how the system can explore this kind of system to transport different things. When we talk about the neurotransmitters, then you get more of that idea about it. Okay. Now, the pump, actually, the transporter are very specific, and it requires for example, in this case, this is the sodium and potassium pump. It requires the presence of the sodium and potassium. They bind into different pockets, and also the ATP. The ATP actually will change the conformation so that the potassium and sodium will transport in a couple of ways. Okay, if missing one will not be able to function. For example, this is the efflux of the sodium as measured by the transporter. If there's no potassium, if you reduce the potassium, the concentration, then the pump will not be functioning as good because it requires both the sodium and potassium together. So basically, what happens is that actually it will bind sodium and potassium, and also the hydrolysis of the ATP, and then that make to the conformational change, and then that actually will move the ions into different directions. Now, same as if you remove the ATP or stop the ATP productions, then the pump will not function either. Okay, so if you add back the ATP, then that will recover. So that's the reason why that actually in the neurons, the energy is so important, because without energy, the pump will not be able to function. And at that time, if that cannot functions, then the gradient of the ions will slowly dissipate. Without the gradient in the ions, that means that the membrane potential cannot establish, and the action potential cannot, pass through. That means that the neurons are not, functioning. If the neuron is not functioning, then that means that the whole nervous system will stop. Okay, so that's the reason why that in terms of energy for our brain to work, that is so important. So for the energy, you need the respirations and also the function of generating the energy for the ATP. Okay. When we talk more about the neurotransmitter, you will see that actually all the activity requires the energy productions. Okay. So here is the example of the pump that we talk about. Is a complicated structure, membrane-bounded structure, in which that with different binding site of the potassium and the sodium, and also the site for the ATP. Okay. Now in this case, there are alpha and the beta subunit as well. Now some of the toxin actually binds to those pumps or transporter that prevent them from functioning. Okay, this is one of the examples. The orban is one of the toxin that actually prevent the pump from working. If the pump is not working, then it will not be able to function. Okay. So it's okay. We talk about the ion channels and transporters. The ion channels and transporters are some of the important molecules that in the nervous system, and any drugs that act on those transporter or ion channels can modulate the activity of the neurons or transmission of the signals. And that's also a lot of drugs how that actually prevent, for example, prevent the sensation of the pain. One of the examples is that, those anesthetics, if that block the transmission of the action potential, then you will not feel anything. Those are examples of how those drugs can act on the channels and then prevent the transmission of the nervous impulse, and then so that how the doctors can perform certain procedure. Now we will have a little bit more details when we talk about those neurotransmitters. Is it okay? Okay. Let's go for a little bit more the structural and functional features of neurons and the nuclear cells. So in the neurons, we'll talk about the structure, the classifications, and the numbers and their function as well. Okay. So here are some of the example of the neurons. Their cell body, You can see that the cell body and then the axon. Now, the neurons are the highly specialized cell. Actually, neurons is the most specialized cell that in our body. You can see that you can see this is the cell body at the retina cell. This is another type of retina cell, different type, the cell body and the dendrite. We'll talk about the axon and dendrite. This one we have seen it before in the cerebella per King cell. Okay, and this is cortical pyramidal cells, in the cortex. Now the neurons are actually so specialized that they cannot go back. Okay, what do I mean by cannot go back? Means that actually once the neurons are differentiated, they cannot divide anymore. So neurons cannot divide actually because they are so specialized. Okay. Now it is so specialized that actually they cannot transform into the tumor cell. So neurons cannot change, mutated and divided into the neurons. Most likely it will, once it is differentiated, the only way either they will live on or they will just die because of different effects. Okay, so that's the reason why that it's so important that during development, when you are still young, your brain is developing, and all the neurons are starting to differentiate it and specialize it, and then into once that you mature, the neural network is actually will be fixed, and that's the reason why that actually when you get older, you're more difficult to learn new things. Okay, just like when you compare, when I compare myself to young people like you, is so I would take more time, for example, to learn how to use the Zoom. I'm sure that all of you, learn how to use the Zoom really, really quick. But for us, I mean, it takes some time because this is something new to us, But nowadays, I mean, I guess everybody knows how to use the Zoom. You cannot survive without using the Zoom. Okay. Okay. The neuron is actually the fundamental unit of the nervous system. They contain, 4 distinct regions, and they carry out the specialized functions. Okay. Now, mainly the cell body. Okay, and then the neurites. Neurites are the outgrowth of the neurons. Now they include the dendrite, the axon, and the terminals. Okay, so the cell body neurites will include the dendrite, axons, and terminals. So here is some of the example of the nerve cell. So this is the nerve cell usually with a big nucleus and a lot of endoplasmic reticulum, mitochondria, and this is the axon. Now, the mitochondria is very important because of the energy requirement in the neurons are very high. So, so that requires the neurons to with the mitochondria to produce the energy. Okay. Now, the neurons can only use the glucose as the major energy source. Okay. The other, if you still remember the biochemistry, what else apart from the glucose can be used? Lipids, yes, but lipids has to be used in a special way. Protein, yes, but protein also needs to be So in the neurons, usually it uses the glucose, and another form of energy that can be used is the energy production. You still remember the ketone bodies? So that can put it back. Yes, ketone bodies. Yes, yes. So that's the energy source that the neurons can use. But majorly it is the glucose. Now, glucose also are very important for neurons when we talk about the neurotransmitters. Some of the neurotransmitters needs to be synthesized with the glucose. Okay. Now, there are also a Golgi apparatus, because the neurons are actually actively doing a lot of the protein productions. Okay, that's also the reason why that in the neurons that as you sometimes you see that the nasal body is the one that actually with the messenger RNA and then attached to the ribosome and produce a lot of proteins. Okay, so the neurons actually you see that it's specialized, but actually is actively producing protein and also degrading protein as well. Okay, so let's look at those. You can see that corresponding to the figure. Now, figure B, that's the axon. You see that's the axon over here, and you can see that actually wrap around is the myelin. Okay. Now let's look at C is the synaptic endings. Now you can see that this is actually the synapse. This is the really the gap. So when we talk about the synapse structure, then we'll talk about it in more detail. Now you see that those small vesicles, these are the electron microscope pictures. You can see that actually those are vesicles that contain the neurotransmitters. When we talk about those neurotransmitters and how that synapse is actually release the neurotransmitter, then we do have more of those. Talk about those. So here is those vesicles. Now here is the axon cut, in those. And you can see that this is the cut, and you can see that those are the myelin. Okay, correspond to this in the D, cut in this side. Okay, so those are the axon, and those are the myelin that wrap around. Okay, so here is the dendrite. Okay, the E is the dendrite. Dendrite is the one that actually sending informations to the neurons. Okay, now here is the cell body, so-called the soma. So the whole cell body is called the soma. And you can see that there's a big nucleus. And this is here, this G, okay, the axon, the look of warrior. So here is a look, and then here is the myelin, wrapping around. Okay. So those are some of the structure of the neuron. So you can see that actually the neurons are actually pretty complicated structure, and they also contain different morphology as well. Now, this is a structure of a typical mammalian neurons. Okay, so here is the axon. So axon send out the action potential, and then that will synapse with the neurons, either on the cell body or at the dendrite. So the dendrite actually receives all the informations. Okay. Now, after receiving all the informations, that will depend on the changing the membrane potential, and if that is strong enough, then at the axon hillock, this is the axon hillock, will generate the initial action potential. Okay, the action potential will transmit through the axon, going all one direction. You still remember all or nothing. So the all or nothing effect is generated in the axon hillock. Okay, and then transmitted through down, all the way down. okay, to the terminals. Now, in this case, this is motor neurons. So if you still remember, the motor neurons will come from the central nervous system, from the spinal cord, and then going out, and then branched, and then to the muscle. Now, the terminals will synapse with the motor endpoint of the muscle, and then that will cause the contractions. Okay. Okay. One thing that actually is kind of special is that, you know, within the central nervous system, the neurons are being myelinated by oligodendrocyte. Okay. Now, however, once they come out to the peripheral, that will be myelinated by a swarm cell. The reason behind that is not quite sure, but there's a very big implication on that. One of the implications is that actually in the axon or the in the central nervous system, once it is broken, it cannot be generated. Okay. Now it is different in the peripheral. In the peripheral, even the axon can, if this is damaged, that can still be generated and then go back to the connections. Okay. So that's also the reason why that if people sustain from a spinal cord injury, they will be paralyzed. Okay. Then those fiber tract cannot be regenerated. Okay. There's a very high intense research on why, you know, how that, if there's any way to promote the regeneration. So when you heard about the news, you know, a lot of people, some of the people, when they sustain a spinal cord injury, they will be paralyzed and then they cannot recover. Okay. But in some of the other cases, you might heard about, you know, particular those people being attacked by the knife. If that damage the peripheral nerve, they can still regenerate, but it takes a long time to grow because of, you know, the neurons, those fiber tract is actually quite long. Okay, this is one of the very important differences. The peripherals actually is managed by the swarm cell in the central nervous system that's managed by the oligodendrocyte. Okay. Now, the neurons in the cell body also contain the organs that you might have studied before. Let's look at it. Some of the dendrite. This is the dendrite. Dendrite receive all the information from different. So for one neuron, usually it will communicate or in touch with a lot of different other neurons. So that's the network. Okay. It is not just synapsing or communicating just with one neuron. Actually, one neuron will communicate with different neurons. So usually they receive it from the dendrite or the cell body. Okay. So when we talk about the synapse, then we will have a more detailed discussion on those. Now, in the neurons, in the cell body, there are Golgi apparatus and also the ER, and as I mentioned about, you know, some of the so-called nasal substance. Why it's called nasal substance? Because those are actually can sting by so-called nasal sting. Okay, that's one of the special sting that you can distinguish neurons from the PSL. Okay. Now there are also a lot of those neurofilaments or microtubules. So the neurons is not just like a web of the cell. Actually, there are a lot of cytoskeleton, and one of those is the neurofilament. Particularly important is that the axon or dendrite actually contain the neurofilament that hold up the structure. Now, in some of the cases, some of the diseases, for example, in Alzheimer's, some of those neurofilament actually having problems, and then will lead to the development of the Alzheimer's disease. When we talk about the Alzheimer's disease, then you'll have more information about that. Okay. As I mentioned, the neurons actually specialized to maintain high rates of protein synthesis. Okay. Because of now in the cell, not just in neurons, in all the cell body, proteins are being produced and degraded all the time. It's like a cycle. So there's some people saying that, 1 month before you are totally different because of a lot of protein has been produced and degraded. Okay, so can you tell why that the cell needs to produce and degrade the protein all the time? Neurotransmitters, another thing. Signal control are big codes. So why we need to produce the protein and then degrade it again? Misfolding, good. Control the cycle, good. Yes. So yes. Now, two things. One is that proteins not when you produce, it is not can last forever. It will be damaged. Yes, proteins can be damaged as well. Oxidations or damage, misfolding. So those need to removed. Otherwise, it will impair the cell metabolism. Now, another reason is because one of you talk about the cycle. Now, if the proteins is always there, then you cannot control, you cannot change. It's only that if the proteins that subscribe to a cycle, then the protein, the cell structure can change. One of the example is cell cycle. Yes, control the cycle. Okay, if this is always there, you cannot change. Okay, so that's the same thing that actually those neurofilaments are being made and then removed and made again. Okay, so that in case that the neurons needs to change, then that can be changed. Now you ask why the neurons needs to be changed. For example, the formation of the memory that actually required changes in actually the physical changes in some of the structure. Okay, then that required changes of the protein productions. Okay, so that's one of the reasons for that. So very good, actually. Let's go. Now, so we talk about the cell body. So here is the cell body. And now we talk about the neurite. Neurite is the outgrowth of the neurons. There are three types of major types of the neurite: the dendrite, the axon, and the terminals. So here is an example of those different neurons. So motor neurons, multipolar interneurons. Okay, when we talk about the spinal cord, the reflex, we saw those multipolar interneurons. And the most common one that we know about is the motor neurons, okay, in the spinal cord. So we have the long axon and then synapsed with the muscle. So here is a dendrite. The dendrites receive input from different neurons. And then the axon, so this is axon hillock. Once that we receive enough information to generate one action potential, then that will generate the action potential and then down all the way to the terminal. Okay. Here is the sensory neuron, same. Sensory neurons are a little bit special in which that the cell body in the dorsal root ganglia, and then that actually will synapse back to the different neurons, either to the motor neurons or interneurons. Okay. Now here is the dendrite, receive information, and here's the axon, and then there's this terminal as well. So they're also all of them are specialized. Okay, so those are the neurites. So the dendrites, as I mentioned, is a large extension of the cell body and then receive most of the synaptic input. So they get all the information from other neurons. So all those information, or actually is the changes in the ions, because they have the channels, either activated or when we talk about the neurotransmitter, we will have a more detailed discussion on those. And what happens is that the dendrites or the cell body receive the neurotransmitters or other signals, and then the whole cell body will change in the membrane potential. If that is high enough, then the axon hillock will fire the action potential. Okay. Now, action hillock, just a little bit more information for you. There are a group of different channels that to facilitate the initiation of the action potential. And again, the action potential is encoded by the frequency, not the amplitude. Okay, so higher the frequency, stronger will be the signal transmitted to the terminals. Okay, axon is arising from the axon hillock, as we mentioned about. That's the specialized in the transmission of the action potential. Okay, now terminals form the so-called P-synaptic components of the synapse. So synapse, remember, synapse is the connecting point between the neurons. So the axon will synapse with the dendrite or the cell body. Okay. When we talk about the synapse, we will have a more detailed discussion about how the structure. So the synapse actually is a very specialized structure in the neurons that modulate the activity. Okay. So actually, it's very simple in which that we mainly talk about the structure of it. Now, we always like to classify different types of cells. So we can classify by neurons by their morphology or functions or the neurotransmitters. Okay, so morphology, function, or neurotransmitters, they secreted. Now, each classifications will have their own advantage and disadvantage. So we'll just look at it how that is being. Now, for example, we can classify that by morphology. So, for example, 1, 2, or 3 or more neurons. So that can be unipolar. That's in the, for example, in the sensory neurons. Bipolar. Some of those neurons that you see that in the retina, or multipolar. So with multiple dendrites and together with the axons. So that actually is classified according to the dendritic tree, or whether they have more spines or links. That is actually one of the good way to classify that, the neurons. So because the how they functions will require they have different kind of morphology. Okay. Now, functional. So you can divide it by functional. For example, sensory neurons, which is in the detections of the stimuli. I don't think that we have the time that to look at how the sensory. For example, sensory neurons actually very special. They can sense temperature, pressure, or pain. So all those are different. Okay, even for temperature, some of those sense that light temperature changes. Some sense high temperature changes. Okay, motor neurons, the one that actually control the contraction of the muscle, and the new interneurons as well. That for example, in the reflex, one of the most simple reflex is the sensory neurons. Synapsed with the motor neurons, but usually more of the cases, if you still remember in the anatomy, actually it involves the interneurons that can modulate the activity. Okay. The classification based on the neurotransmitters. Now, it is the one that actually more useful in the sense to distinguish different type of neurons. Now, because each neurotransmitter, when we talk about it, it will need a specific metabolic pathway to produce it, to store it, to metabolize it. So that means that there's a special enzymes or protein molecules or transporter in the cell specific for the metabolism. Okay, when we talk about the neurotransmitter, you will see, so the production, the storage, the release, and the breakdown of it. So those are specialized pathway. So that means that in those neurons, they will have specialized enzyme for those purpose. Okay, so so that's how you can classify them. So when we talk about those neurotransmitters, you will see that actually one of the examples is the cholinergic. So in the motor neurons, it use the neurotransmitter, which is acetylcholine. So those motor neurons, you have the enzyme, specifies an enzyme to generate, to produce the acetylcholine. Okay, so usually those neurons we call cholinergic. So that means that they use the particular acetylcholine as their particular neurotransmitters. Okay, so that's the reason why we call it cholinergic. Okay. So in the human nervous system actually contain around 300 to 500 million billions, or 300 to 500 billion of neurons. So you see that actually there are a lot of neurons in the cell, and the density actually is quite constant across the cerebral cortex in different mammals. So that's the reason why that the smaller brains have fewer neurons, and bigger brains have bigger brains have more neurons. Okay, that's usually the case. That's the reason why that in the elephant the brain is much bigger, and then actually the brain is actually they have more neurons. Okay. Okay. Before I continue with this, just to let you know a little bit of things that I'll post that slide after we finish the class. I just want to mention about the day of the midterm, and also the time of day of the midterm, and also the assignments. Let me check. Let me stop this one first. Okay, so the assignment due date is the October 23rd. Now, actually, the assignment actually is, I just need you to watch a video. So you click on this link, then you can get to the website in which that is a Nobel Prize talk. I want you to watch it, and it's about the structure and the mechanism of the potassium ion channels. Okay, so the idea is I just want you to watch and then know about how the potassium channels are being selective, and I just need you to write a one-page summary, just one page. Okay, and then submit to the campus. I will open the campus for your submissions. Okay. I will not, I just want you to watch the major target is for you to watch the video. Okay. Yeah. If you submit a summary, then you get 5 points. Okay. If you don't submit, then you don't get the 5 points. Okay. Is it okay? 5%. So this is the first assignment. Is it okay? Anyone want to ask anything about the assignment? Now, the midterm will be on 19th of October, okay? I will give you more information. Okay, actually, is this a bonus or this is required, not a bonus? So remember, in the course outline, we have 10% for the assignments. We have 2 assignments. So this is the first one, which is 5%. If you don't do it, then you don't get that 5%. It's not bonus, okay? So remember that I will set a date, midnight of the 23rd of October. That will be the deadline, okay? And the midterm will be October 19th. As midterm, I will tell you a bit more details later. I haven't finalized it yet, but it will be online, as mentioned from the university. Is it okay? So I'll upload this to the campus, and then we'll talk about the midterm, more details about the midterm once that I finalize the midterm questions. Is it okay? Any more questions? And in the next class, then we will discuss about the project as well, because the edge-up periods have just finished. As I mentioned, is there's no one of you asked like more information. As I mentioned, is you just watch the video and then do a one-page summary uploaded to the canvas, and then that will be. I need to the coverage. I will give you a more detail about the coverage when we get close, maybe in October, beginning of October. Okay. We are still in September. Okay, so we'll go back to the class. Just a moment. I have some. Okay. Okay, so we talk about the neurons, and then important cells in the nervous system is the neuroglia cells. Okay, neuroglia cells is another one. Now, neuroglia cells are the mainly doing the supportive functions. For example, one of the important aspect is the neuroglia cell produce the myelin that insulate the neurons from each other. The midterm, I will talk about the midterm, more details about midterm later. Okay, it's still early. to talk about whether it's closed or open book. Is it okay? Yes, I will mention about the midterm, more information about the midterm later. Okay. The glia cell produce the myelin, for example, and the neurons, the glia cells are different from the neurons in which that the neurons actually they can the glia cells they can proliferate and they can divide compared to neurons. Neurons, once it differentiates, it cannot divide anymore. Okay. The glia cells do not actually produce neurotransmitters, and also they don't have the synapse. But they are also the glia cells are actually very important in the transmission of the neurotransmitters. Okay, when we talk about those neurotransmitter pathway, then you see that actually the glia cell are also playing an important role in the neurotransmissions. Now, as I mentioned about, the neurons cannot be divided and divide again once there is this differentiation. That's the reason that maybe you heard about that in the their brain tumor. Now, the brain tumors are mostly gliomas. Okay, it's actually from the new glia cells developed. And in the brain tumors are usually actually is quite lethal, and then most often it is not able to cure because that it's very difficult to remove it. First of all, is if you remove it, it also removes part of the brain tissues, which is not easy. And also that if you remove any brain tissues, then that will affect the patients as well. So usually the gliomas will be easily relapsed. Now you can see that actually new glia cells are actually 5 to 10 times more than neurons, but they occupy similar space. So that means that in the nervous system, the brain actually half of it is neurons, half of it is actually new glia cells. Okay. So there are different types of glia cells, major type. For example, epidermal cells, astrocyte, myelin-producing glia cell, including the oligodendrocyte, or the oligodendroglia, or swarm cells. So remember, the swarm cells are actually peripheral nervous system. The oligodendrocytes are actually in the central nervous system. Microglia cell, those are related to the inflammation. Satellite cells. So we will talk about each of those. So here is the astrocyte. Okay, this is the oligodendrocyte and microglia cells. You see that they have a lot of pulses that pulses out. Okay, those are the staining of the astrocyte in the brain. So it's like a star shape. Okay. That's another pictures, is a electron microscope pictures. There are pulses. This is the one that actually the warrior over here, and those are the myelin. Okay, wrap around the axon, the oligodendroglia cell. Now those are actually a microglia cell. Now microglia cell are just like the immune cell that actually activated when there is a damage or when there's a pathogens. Now, actually, in the brain, if there's any inflammation, that is not a good thing because that usually will damage the tissue. Now, in all cases, inflammation is not necessarily good for you because that when the immune system is activated, it tries to destroy the pathogens. That's the time that actually that might have the collateral damages, which also might damage the other tissue as well. So that's the reason why that, for example, in the case of pneumonia, in SARS, that is so damaging to the lung. because once the lung is damaged, it cannot perform the gases exchange. That actually is lethal. So you can see that what happens to Hong Kong or around the world for the SARS virus attacking the lung tissue and causing the pneumonia. That's the reason why it is so lethal. Okay. Here is some of the example of how the shapes of the glial cells. So the astrocyte actually help to form the blood-brain barrier. If you still remember, it's not forming the blood-brain barrier, but actually signal the epithelial cells of the capillary to form the tight junctions. Okay, so those are from the astrocyte. Radial glial cells that actually help to develop in the axon, and also the oligodendrocyte wrap around the axon, forming the myelin sheath. In the case of the peripheral system, that's the swarm cell, okay, wrap around. So you see that all the wrapping around is actually a lipid and heavy in the lipid. So that's the reason why that, there's a so-called white matter, because all those are making the color looks white, actually just like fat, that's the reason, okay. This is a microglia cell, in which that, as I mentioned about, those microglia cells are the one that are involved in the inflammations of the brain, if that happens, okay, during the damage, or either when there's accidents that damage the brain tissues, or when there's a inflammation in the brain tissue, okay. Epidermal cells are actually one that actually secrete the in the plexus that produces cerebral spinal fluid, the CSF. Some of those are also forming the leaky blood-brain barrier. So in some of the part of the blood-brain barrier actually is more leaky than the other one. It all depends on the site of it. Okay. So astrocytes are large, numbered, star-shaped glial cell. They usually have a very long pulses, tap the feet. They form up the take up the space in the nervous system. So the astrocytes actually have two classifications, two morphological varieties. Fibrous astrocyte, usually in the white matter, and protoplasmic astrocyte usually is in the gray matter. So it will occupy the space that actually between the neurons, okay? So the astrocyte actually will fill the space of the neuron that is not occupied by the neurons or blood vessels, okay? They also cover the synapses and then also contact with the capillary endothelia to encourage the formation of the blood-brain barrier. So they're also in the pia matter, where they form in the limiting the glial membrane as well. Okay. Now the function of it is actually homeostatic regulation of the extracellular potassium ion. Okay, and also in the removal of the neurotransmitter from the synaptic cleft. Okay, when we talk about the neurotransmitters pathway, you'll see that actually the astrocyte will also functions helping the synaptic transmission. As when we talk about the neurotransmitter, you will see that actually once the neurotransmitters released, it needs to be removed as soon as possible. Still remember, why is it the case? Anyone? So why the neurotransmitter needs to remove as soon as possible?
To avoid like prolonged activation, like
yes.
This should stop.
Yes.
After.
Yes, very good. The removal, if this is not removed as soon as possible, once the transmission is done, then the second transmission cannot occur. Okay. There's also the reason behind it when we talk about some of the drugs actually is targeting this pathway. If you prevent the neurotransmitter from removing, for example, in the stimulation of the muscle transmission, the muscle will contract all the time. Then that means that if the muscle continues to contract, that will become paralyzed. Okay, that's also some of the drugs actually targeting this pathway. When we talk about those, then you will see that also the astrocyte also involved in the regulation of the supply of the glucose to the neurons. Okay, it's helping the metabolism of the glucose to the neurons. That's actually very important. One is the energy production. Another one, also very important, is the generations of some of the neurotransmitters. For example, in the glutamate, that's one of the important neurotransmitter in the brain. And then when we talk about the pathway, you'll see that why actually it is so important for the control of the regulation and production of it. And if you still remember from the biochemistry from Professor Cole, then you see that how that actually is generated. You think about it, how the glutamate is being generated in the Krebs cycle. Okay. It also helping to form the tight junction in the blood-brain barrier. Still remember the astrocyte actually secretes some of the special signal to the capillary epithelia to form the tight junctions to form the blood-brain barrier. Okay. Oligodendrocyte. So oligodendrocyte in the central nervous system and also the swarm cell in the peripheral nervous system, both are important in the formation of the myelin sheet at the axon. In the central nervous system, there will be oligodendrocyte. In the peripheral, there will be a swarm cell. If you still remember, we talked about the multiple sclerosis. In the case of multiple sclerosis, is the immune system attack the myelin sheet, and then result in the short circuiting of those axons, and then those are the reason why that those patients suffering from it will have a weakness in the muscle, or they have problems in the movement because of the problems in the transmission of the signal. Now, microglia cell, that's the types of the phagocytic immune cells in the bone marrow that believe that derived from the bone marrow, from the monocyte, similar origins. Okay. It is usually the immune system are not activated in the nervous system, the central nervous system, because once it is activated, that's what believed to be the case in the multiple sclerosis. For some reason, is activated, and actually the nervous system is not, the immune cell is not recognizing the components, and therefore attacking the myelin sheath. Okay, so that's believed to be the cause of the multiple sclerosis. That's also is the reason behind that. The cause of that disease is not completely known, but it's believed either there's a inherited or is caused by some of the bacteria or virus that actually infect the central nervous system and cause the activation of the immune system in the central nervous system. Okay. Now, as I mentioned, that actually that can remove the damage the neurons or tissue in the health or during the healthy or disease state. Okay, or we repair their function. So that's the phagocytic cells in the nervous system, in the central nervous system, that the microglia cell. Okay. Now there's also so-called the satellite cell. Now that actually present in the peripheral nervous system, in the ganglia. Now, in the ganglia, it also is not just as I mentioned, the ganglia is a group of neurons in the peripheral. For example, dorsal root ganglia, in the dorsal root that holds the sensory neurons body, or in the sympathetic ganglia, or the parasympathetic ganglia. Now, those ganglia actually not just contain the neurons, but they also contain some of the satellite cells. They are similarly as the glia cell. Okay, even in the ganglia, they're also half-half. Okay, so that forming the cellular cell actually helping the neurons in the ganglia to function. Similar in the central nervous system. Okay. Okay, so we finished today's about the structure and function of neurons, and also talk about the channel and transporter. So any questions?
Hello, Professor.
Yes.
I actually have a very principal questions. It's basically our brains initiate some of the electrical signals to every part of the body, but the signal is basically the same. And how come our body know what to do? Like, I want to move my hand, but how come like I receive it to my hand, then my hand would move? Is it because like the cell has already differentiated and then they would serve their functions, or there are other reasons?
Well, I think that is a little bit more complicated. If you think about your movement of your hand, so you have to think about your thinking and then initiate a signal movement, and then going all the way to the spinal cord, and then you control those motor neurons. So it's not a very simple way. It's like, oh, the brain like send a signal to the hand and then they move. Actually, is a process that involve the initiation of your thinking, and then also initiate forming the movement. Okay, so it's not just that simple. It's just one simple process. Okay, when you look at it more, then you'll see. So that's the reason when I talk about even for initiation of a sentence, it's very complicated. You have to think about what you want to say, and then generate a sentence, and then that send that information to the motor neurons to control your mouth and your tongue and how you pronounce, or also your vocal cord as well. Okay, so but it's very good. You start to think about it, then that actually is a more complicated process. So how do you let me see if there's some. Is the cerebellum part of the brain stem according to the previous? Yeah, you can say that it is part of the brain stem. Yes. Okay. How do you distinguish between dendrites and axon if you are looking at a neurons under a microscope? Now, actually, the dendrite and the axon, they have different markers. Okay, but when we talk about the synapse, then we can talk more about it. Well, actually that's the case as well. Okay, the dendritic spines. So cell cycle is one of the reasons why that the proteins are being degraded all the time. Okay, for example, if the proteins are being degraded all the time, then for example, the cell cycle will not initiate to divide. But if somehow the protein is being not degraded, then that can signal the cell to, for example, divide. Okay. Same thing, the protein structure and the control of cell cycle is same for the other cells. So there are some studies which saying that the neurons will not degrade, will divide because of the some of the proteins are not being degraded, or some of those are always being degraded. Is it okay? Any more questions? Yeah, when you think of it, actually, the things that we are discussing in this class is actually very simple, and some of those are, if you ask me, I cannot explain to you 100%. For example, one of your students asked about how the movement of the hand is generated. I mean, the whole process is actually very complicated, and particularly when we have time to look at some of the diseases, then you see that actually sometimes something happens, and then you see the outcome of the disease, and you see that some of those systems are eroded. For example, in the movement. Okay. Okay. Is there any questions? If not, that we will end here, and then I will see you guys on Wednesday. Okay. 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 Bye bye. Okay. Thank you. Bye bye.